much. Thank you very much for the opportunity to come and talk about the idea of large prospective studies. I find myself in a slightly odd situation in that I kind of half agree with the title of the meeting. I've been very keen on the idea of big data, but actually I'm a fan of imprecision medicine in the sense that what I would like is to have the best average treatment for people rather than precise treatment for a small number of people. But at least I'm half in agreement. So, what are the advantages of setting up some very large prospective cohorts where you collect information from questions, measurements, biological samples, and then wait a long time? And I think from a genetic perspective, probably not a lot of advantage. If you're only interested in genes, then case control studies make a lot of sense. But if you're interested in the complex interplay between lots of different kinds of risk factors, gene, environment, lifestyle, interactions, then those factors that can be themselves changed by disease need to be assessed well before the disease develops. I mean, after all, one of the best ways to get people to stop smoking is to encourage them to have a heart attack. That will then get their hearts and minds. So we need to do these studies where I can see the slides. Can you see the slides? Yeah. So we need to do these studies where you measure everything beforehand. The advantage of these big cohorts is you can also compare like with like to a much greater extent because you're looking within the same kind of population. And they're holistic. So rather than looking at one particular risk factor or looking at one particular disease, they're very, very cost-effective in the long term. You can look at the interaction between lots of different risk factors on a single disease or a single risk factor on lots of different diseases. But they need to be very large. And the reason for that is that although everybody in the long run will be informative about one or other disease, of course, only a small proportion, or a relatively small proportion, will be informative in that they will develop any particular disease. So to have enough people that develop a particular disease, you need to start out with a very large number. And to give you some idea of that, I've worked with a colleague called Sarah Lewington who brought together about 60 prospective cohorts with about 12 years of follow-up with 1 million people. And then I said to her, select randomly out of that the first 5,000 individuals, which she did, and that's shown on this graph. And it's looking at the interaction between two risk factors, blood pressure and age, on the risk of death from coronary artery disease. So two risk factors that have big effects, and their effect on a common condition. This is 5,000 people. So this is the scale of the Framingham study. This is the basis on which your doctor will tell you that your blood pressure is too high and it should be lowered. And if you look at it, if you look at the plots with the confidence intervals around the plots, what you kind of see is a blur. You see a cloud vaguely going up, but you can't really determine the shape of the association. Go tenfold bigger, and you start to get better shape, better idea of the association of these two interactions on a common disease, but still not the kind of clarity that you'd really like if it was you that was trying to determine whether or not you should have your blood pressure lowered. Is there a J-shaped curve in the younger ages? Is it flat at the higher blood pressures in the older ages? Go up to 500,000 people, and it's like having a microscope that you bring into focus. You get these beautiful log linear associations. They're flatter at older age, so in relative terms, the interaction is weaker at older age. But of course, the absolute risks are much higher at older age, and these are plotted on a doubling scale, so the absolute differences for any blood pressure difference are much bigger at older age, so the absolute interaction is greater at older age, so you can discriminate. But this is 500,000 people, 12 years of follow-up, two strong risk factors on a common disease. So if we're interested in effects which are more moderate on less common conditions, you can see why we need to be thinking in terms of millions of people. The other thing we need to think about is that the UK or the US is not a normal population. We're people who live in a very abnormal way. So if we want to understand the full effects of normal exposures, we need to be studying in a variety of different populations where we have perhaps more normality. And so heterogeneity is valuable. How do we build a series of studies that enhance the heterogeneity? Bear in mind that diseases that are rare in one population can be common in another. That's good news, and it suggests that they can be prevented. But it's also important to use that to, again, extend the range of diseases that we can study. And my point about exposures, if we want to look at, say, adiposity, then we want to have people who live in a population where there's a lot of it, and people who live in a population where there's very little. So it's heterogeneity in these prospective cohorts that is our friend, and we should not be trying to establish representative cohorts, because representative of what? If we get a representative collection now and then follow them long term, populations will change, so they won't be representative of the population we're trying to apply them to. Here's an example of the kind of heterogeneity of risk of disease levels. If you are interested in stroke, study it in China, where it's common. If you're interested in various kinds of cancers, study in China rather than the UK or the US. And even within a population, say like China, there's very large unexplained variation in the levels of different diseases. So these are data from many different villages around rural China, looking at the death rates from different kinds of disease, COPD, esophagus cancer, stroke, coronary artery disease. And you can see that even within this population, both in men and women, very wide heterogeneity in the disease rates. So my colleague in Oxford, Zhengming Chen, working with the Chinese Academy of Medical Sciences, set up at the beginning of this century a cohort of 500,000 people with questions, measurements, biological samples, linked it in to the health insurance scheme in China, which is a billing system, so it's very accurate about all the things that happen, and is now following that cohort up. And I just wanted to kind of show some playful, interesting findings that come out in that. So here we look at the association of markers of adiposity, could be BMI or estimated fat, with blood pressure. We see the expected associations, more adiposity, higher blood pressure. If we look at the associations of blood pressure with the risk of coronary artery disease, but also here the risk of stroke, and because stroke is so common, it's the CT scans done immediately on admission, so we get very good quality data on the type of stroke. We can look at ischemic and large numbers of hemorrhagic strokes. We see the expected association of blood pressure, higher risk of both ischemic and hemorrhagic stroke. Actually, slightly stronger association with hemorrhagic stroke. And therefore, QED, higher adiposity, higher blood pressure, higher risk of stroke, and that's what we see for ischemic stroke. But interestingly, not for hemorrhagic stroke. Higher BMI, higher blood pressure, not associated with a higher risk of hemorrhagic stroke. What is the protective effect that is counteracting that increase in blood pressure associated with increases in BMI? And I don't know the answer, but it's nice to get very reliable questions that one could then address. Looking at range of exposure, taking the example of obesity, we can go to the other extreme and go to Mexico. And again, a colleague of mine, Jonathan Emberson, working with the Mexican Ministry of Health, with support from the Wellcome Trust in the UK, have set up a cohort of about 150,000 people in Mexico City. And as you can see from here, very high rates of obesity, both in men and women in middle age. And associated with that, very high rates of diabetes, about 20% of men and women in middle age with diabetes compared with less than 10% in the US or in the UK. And in that setting, extraordinarily large relative risks of death from, in particular, renal disease, but also cardiovascular and infectious diseases associated with that high rate of diabetes. Much stronger associations than are seen in the West, and particularly strong associations for renal disease. There turn out to be two reasons for that. One is that if diabetes leads to chronic kidney disease and to renal failure, then there isn't the backup for many people of dialysis and renal transplant. It is a death sentence. The second is, why is it more powerfully associated than the case in other populations? And it turns out that that relates to the way in which the diabetes is managed. But if we look at the absolute impact in this paper that was published in the New England Journal in December 2016, about 40% of the deaths in middle age are being caused by diabetes in this Mexican population, and almost all the renal deaths are a consequence of obesity leading to diabetes. And having measured HbA1c in this population, what we find is that the HbA1c levels are much higher in the diabetics than would be the case in a Western population with good diabetes control. So we were identifying a major health problem that had been going unnoticed, which is actually solvable by better control. So all the people with diabetes were known to have diabetes. We found very few undiagnosed diabetics, but they weren't being well controlled, and they weren't getting other cardiovascular protective treatments. So within a week or so of these results becoming available, the Mexican Ministry of Health across Mexico had implemented public health strategies. So this stuff can be translated very quickly. And following on from those studies, with the support of the Medical Research Council, our equivalent of your NIH, and the Wellcome Trust charity, in 2006 to 2010, we recruited 500,000 people through the National Health Service with lots of questions, measurements, and samples, and have been following those individuals up through linkage to the health record systems in the UK. We've been looking at ways of enhancing that characterization of the participants, and we're very interested in ideas coming from anywhere about how we could use particularly remote approaches or monitoring devices to characterize even more finely the participants' phenotype. We've enhanced, for example, the food frequency data with internet diaries, and we're doing more detailed cognitive assessments, not only to assess their baseline cognition, but to use that as a follow-up for cognitive decline, going beyond what you can get from health record systems. Doing a standard panel of assays, about 35 different assays, along with hematology, and making those data available. And I should stress that UK Biobank is available to any researcher, academic or commercial, anywhere in the world, for any kind of health-related research. The advantage of doing these whole cohort assays rather than doing case control is, again, in the long run, they're very cost-effective because you can do the assays all in one go, make sure you get very high-quality control right across the whole cohort, and then when you want to study different diseases, you can just pull the data out, and it's a very fast way of making data accessible. It also ensures that we don't deplete the sample. We collected about 55 mils of blood, about 3 tablespoons full of blood in lots of different tubes, but still over the next 5, 10, 15, 20 years, there are lots of things we want to do in those samples. But of course, if you take 500,000 of anything and multiply it by any number of dollars, it'll turn into a lot of money. So to do whole cohort assays is expensive. To genotype all the participants cost about $30 million. To do the 30 to 40 biomarkers we've done has cost around $15 million. But as the data are becoming increasingly valuable, more and more people are thinking about how they can turn the samples into accessible data. And so, for example, with Regeneron, along with a number of other companies, have pulled together a consortium, and they are now exome sequencing all of the cohort, and then those data will be available to all researchers to use. We're also just starting a vanguard pilot phase to whole genome sequence the first 50,000 participants with the intention over the next 3 to 4 years to sequence the remainder of the cohort. So whole genome sequence data would also be available. We've got 100,000 of the participants to wear accelerometers to enhance the questionnaire data about activity. And it's quite interesting how you get interestingly different answers from looking at the associations of various factors with wrist-worn versus questionnaire data. So here is some data from Aidan Doherty, who has turned the accelerometer data into activity data, looking at a Manhattan plot. I guess this is a Manhattan plot in Sydney. Looking at the self-reported activity with the genotype across the whole of the genome. And this is the Manhattan plot with the accelerometry data. And you'll notice, kind of over on the far right, that there is a very strong peak for the association with accelerometry data, which is not seen with the self-report. So they seem to be assessing different aspects of activity and enhancing the data. As I mentioned, all 500,000 people have been participants. Of the participants have been genotyped, and many people here are using those genotyped data. Setka Theresin and his colleagues in the Broad have done a nice study looking at the association of the genotype risk score with coronary artery disease. And interestingly, and I think not really predicted to be the case, they've identified quite a substantial proportion of individuals with a genome score that have an equivalent risk to having being a heterozygote for familial hypercholesterolemia. So about 2.5% who are very high risk based on a genetic score that is over and above the standard risk factor measures. So this becomes an interesting way of identifying in primary prevention high-risk individuals that could benefit from interventions like statins. And the kind of final enhancement that we're in the process of doing at the moment is to image 100,000 of the participants. We've so far imaged about 20,000 with 3T, so a 3 Tesla magnet of MR of the brain, 1.5 Tesla of the body and heart, DEXA of bone, joint, and body, carotid ultrasound, and then a complete repeat of the whole of the baseline questions, measurements, and biological samples. And we're now also working with various people over here to get cardiac rhythm monitoring for a couple of weeks using the kind of patch-like approach in the first 20,000 individuals to try to look at atrial fibrillation in more detail. Again, an interesting result that's currently in bio-review, looking at the associations between the genotype and markers in the brain. And in this brain image, the yellow patches are patches of what would be considered microbleeds, these hypodensities that show iron deposition. And the associations of the genotype with that deposition. And what turns out to be the case is that the 4 SNP variants that are strongly associated in the Manhattan plot are specific to subsections of that yellow patch. And all 4 of those variants are associated with iron transport. So those hypodensities may well not be blood and bleeding. They may well actually be iron deposition itself, misinterpreted as bleeding, and that these SNP variants which are associated with ion transport, are also associated with cognitive function decline and dementia. And that what we're seeing here is possibly a new therapeutic target for dementia relating to ion transport rather than a concern about bleed. So again, an interesting observation. In these large prospective cohorts, the focus is often on phenotyping the participants and then linking into rather dumb health record linkage data, which may have a lot of imprecision in it. And it's certainly the case that one can enhance the phenotyping not only of the participants, but of the health outcomes and get even more precise associations. In the main, the concern is false positives. And missing a few positives will not have very much effect on the associations. But you do really want to be sure that the things you think are the disease really are the disease. And the more you can subdivide the disease into specific subtypes, be it, say, ischemic or hemorrhagic stroke, again, the more specific the associations one would look at. And again, to give an example of that, if we take things like reproductive history and look at its association with different kinds of breast cancer tumors, then the reproductive factors are strongly associated with breast cancer tumors that have hormone receptors on the surface and not with those that don't. So you can imagine that if you couldn't subdivide those breast cancers, you would get a kind of average association, and you would miss this very specific difference. And so in building these cohorts, we need to think not only about the data that we can get and interpret now, but also how we ensure that we can get to other kinds of data that will allow us to enhance the phenotyping of the health outcomes in the long term. Be that image data that we might be able to access to look at, say, stroke in more detail, or tumor samples that we could go to in 5 or 10 years' time and do some new test that allows us to characterize a particular tumor more specifically. So trying to future-proof the cohorts, not only by storing samples from baseline, but also storing data that we don't know what it means, or storing tumor samples we don't know what they mean, that we can go back to in the long term. And so the approach that we're taking in the UK Biobank study is first use health records systems and also self-report with participants to try to ascertain disease case. Then develop approaches to combining that with other more specific data to confirm caseness so that we get rid of false positives. And then look to see whether we can go even further, accessing more precise data to get more specific characterization of the health outcomes. But at the moment, there really are no established approaches to doing this at a very large scale for very many different diseases. So many of us in this area of large prospective cohorts are starting to work together around the approaches of taking a lot of data about health outcomes, using the data to tell you how it should be subdivided, to tell you what a participant's disease is, and get precision and specificity. Finally, it's good to have big cohorts. It's good to have well-characterized cohorts. It's good to have follow-up of those where you get specific information. And it's ideal to do it for a very long time. So colleagues in Oxford used the breast cancer screening system within the National Health Service to recruit 1.3 million women some 20 years or so ago. And they've been looking at dementia in that study. So they've got 35,000 dementia cases already and about 50,000 over the next 5 years. And the thing about dementia is that it changes the way in which you behave. And it does so over a very long time. There's a very long prodrome. In fact, it's about 10 years. So this shows the association, the relative risk of dementia among women who were inactive versus active. And you can see that soon, in a short time before the dementia, there appears to be a very strong association of activity protecting against dementia. But after 10 years, you don't see that association. This is reverse causality. This is dementia causing inactivity over a very long time. And so when you look at the association, and you see in the red box at the bottom, activity is not a protective effect of dementia. It's dementia that is causing the inactivity. And so very long follow-up for diseases with a long prodrome is important. So in conclusion, in these prospective cohorts, to study the full effects of many different types of risk factor on many different types of disease, we need to have several large-scale studies. We need to have millions of people in different populations. We need to work out how to enhance all or a very large proportion of them in terms of characterizing the participants. We need to build those into health record systems that allow us to get long-term follow-up. And finally, we need to make sure that we can present those data to researchers around the world in ways that allow them to understand the data and how they can use it for their research. Thank you very much.
Terrific. Thank you, Rory, for opening up the afternoon. And please, as with the spirit of the morning, come up to the microphone, and I'll try to see you when you get up there. Rory, one of the things that we were talking about backstage is that you've certainly been interested both in the collection of epidemiologic cohorts as well as large-scale clinical trials. And from the clinician's perspective, a lot of what you've talked about is interesting associations. You've chosen your word very carefully. Clinicians are also interested in what makes a difference and what's actionable, and patients are interested in what makes a difference. How are you reconciling this call for real-world evidence as an alternative to randomized clinical trials? And how are you thinking about your own work? Are you doing trials off the backbone of a cohort study, or just other things that you're thinking about in this space?
Yeah, I mean, in population science, there have often been two kind of schools. There's the observational epidemiologist and the people who do randomized trials. And I've tried to be in both schools because I see observational epidemiology as a very good way of generating interesting hypotheses. And I see the randomized trials as a very good way of testing those hypotheses. In a lot of examples, though, a randomized trial turns out to be a very kind of good way of killing your beautiful hypothesis, but be that as it may. And so when one looks at cholesterol lowering, there was for a long period of time a belief that one should lower high cholesterol. But hey, we had average cholesterol, so we were fine. But we had average cholesterols in a population with an abnormal level of coronary heart disease because we truly had abnormal cholesterols. And it was our work in China where we had observational studies showing much lower levels of cholesterol with much lower levels of coronary artery disease that pushed us to do randomized trials of the effects of lowering average cholesterol in high-risk individuals and demonstrating that was beneficial. What I worry about is the idea that they are an either-or. What's happened over the last 20 years is that randomized trials have become much more complex, much more costly, driven by over-regulation and over-interpretation of poorly thought-through regulation. And there are two solutions to that. One is you get rid of the over-interpretation of regulation, which is inappropriate, and that would be my solution. But the solution that's being grabbed at the moment is, well, it's too costly, it's too complex, let's use observational data, let's use so-called real-world data, which is, I think, a brilliant piece of branding. Real-world data sounds real.
How could that not be good?
Yeah. So as a branding exercise, I think it's been very successful. But as a public health exercise, I think it will be a disaster because observational studies have the potential for biases. That you can have the smartest informatic people, the biggest computer, the largest database, and you can get very precise answers, precisely wrong answers. And that's where the magic of randomization is really not understood, that it gets rid of all those potential confounders. So if you're looking for big effects on common on conditions that you wouldn't normally expect, smoking on lung cancer. Why was lung cancer being studied? Because there was this epidemic emerging. It was a huge effect. Observational studies are great, but for moderate effects, or distinguishing between moderate effects and no effect, which is what most treatments are, you need randomized trials.
Yeah, I've taken to, rather than introducing myself as one type of researcher or another, I say I'm a clinical trialist and observational outcomes researcher, which sort of covers the gamut of things. I don't know if anyone's up at the microphone. Yes, in the middle here.
Yeah, Rory, it's Ewan, actually, from Stanford. Thanks for a great talk. I wonder if I can sneak in 2 quick questions. The first one really goes on from the point you were just making, which is how far are we moving along the road towards causality modeling that, say, in economics that has been fairly mature in the clinical observational study world. And the other is more practical. We had a great talk from Eric Dishman this morning about all of us, and I know he mentioned that you've been working very closely together. We know you've been involved from the beginning. What sort of advice have you been giving the American version, if you like, that lessons that you've learned from UK Biobank?
To your second question, my main advice is procrastinate. Do what you have to do now. Think about what you'll have to do in the future, but don't necessarily do it. Because things will change. If we had tried to do all the assays of the samples in UK Biobank as they came in, then it would have cost us a lot more money, and it would have been done worse, because the people who come into a study come in a sequence which is not random. And so what you really want to do is wait until you've got all the people in the study and then pull your samples out in a random order and then do the assays. So you use the study to give you an internal control. There should be no shift over time. So actually, it's better to wait. Cheaper and better quality. And so procrastination and delay, defer until you have to do it. There's no point in having an access policy until you've got something to access. So that was my first thing to that. In terms of your first question on modeling, the trouble with modeling is you can get beautiful models, and they may be right, and they may not be right. And so the question is, can you rely on them? And that is the difference for me between an observational study, which is about association, and a randomized trial where you can do it, where you can test whether that association is causal. Obviously, one can do things like Mendelian randomization with genetics, which perhaps allows you to look at that as well. But again, we're still using the basis of randomization. And I worry that the National Academy of Science at the moment has got a 3-part series on this, and I think they're getting it wrong. I think they are not thinking about... People don't understand what randomization is really about, other than it's the way to get a paper published in a journal. You do a randomized trial. They don't actually understand the power of randomization inherently.
Rory, I want to, and please come up to the microphones, but I want to get to a topic that Mark Cullen's group was going through this morning, and that is some of the disparities we see both in clinical care and in research. And you made a comment in your presentation on, I wrote it down because I loved the phrase, heterogeneity is our friend. How in the UK Biobank are you assuring that the population is as heterogeneous as you can make it in the UK, particularly from a socioeconomic perspective?
Yeah, so the difference between heterogeneity and representativeness is that you would like to have large enough numbers of people in the different groups that would allow you to compare the associations that you see between those different groups. And so if you think about it, representativeness, if you're interested in, say, minority groups, would mean that you would have a minority in your study. So if you're really serious about generalizability, What you actually need is oversampling of minority groups such that you have enough of those. Very difficult to do. They're often hard to reach groups. And then you think, well, what's the smart way of doing that? How would I go, for example, about getting a large number of Chinese? I might go and do my study in China rather than trying to recruit Chinese in America or Hispanics. So that's where I think setting up the studies in those populations where they're not minority groups and getting large enough numbers, getting heterogeneity. Within UK Biobank, we got exactly the proportions of ethnicity in the UK population. So it
looks like the UK.
So it looks like the UK in that respect, and it's worthless for studying the effects of ethnicity because the proportions who are not Caucasian are so small. But for socioeconomics, much harder to get lower socioeconomic groups. It's much harder to get young males from lower socioeconomic groups. So the things we did were around recruiting at weekends, recruiting in evenings, getting companies to allow their staff to join, locating assessment centres in places where we could get large numbers of different kinds of places. You know, central London, you'll get higher socioeconomic. You go to Nottingham, Birmingham, you can get larger numbers of socioeconomic, more deprived groups. But the key thing is to have large enough numbers from the different groupings.
Along that topic, one of the things that the Oxford group in the trials world has done so well is the trialist collaborations, antiplatelet therapy, cholesterol, and the power of that is obviously the larger numbers. You're talking about partnering with different groups from around the globe. Are you taking the same approach that you took in the trials world, which is that you had to have the data come into a common data structure, or are you doing the analysis separately and bringing the estimates in a common way? And why doing it differently?
Well, with UK Biobank, what we're doing, our job is to build a resource that others can take the data from and combine with other studies. So, I mean, and I think that's probably the way that one would go. The point I do want to make, though, is that, and it's a little bit counter to what was said in the previous session, that you can have precision and you can have scale. And the idea, I think, that was referred to, that precision is more important than scale, I think is wrong. I think the important thing is to have the right balance between precision and scale. So if you go back to the early days of genetics and case control studies with genotyping, there were a lot of case control studies that came out. One would be positive, then the next one wouldn't be. And after a while, Nature Genetics said, enough, I don't want any of these. And people were spending enormous amounts of time defining very precisely, what is coronary artery disease? And one study would define it very precisely in one way, another study very precisely in another way. And we got all these variable results, or apparently variable results. And what they were, in my view, was a bit of winner's curse, the falsely positive result, a bit of data that had been tortured and had lied and misled people. And then what happened is everybody took all their studies with all their different definitions of what is coronary artery disease, combine them together, and suddenly out came these signals. So the undue focus on precision at the expense of scale got the wrong answer.
Can be misleading.
Yeah.
Let's finish up maybe with a comment that also got raised this morning about, I think somebody asked the question about what are you going to tell all the PhDs, the grad students, the medical students about what to do in the future. You're a physician epidemiologist. What do you want to tell, from a training perspective, our clinicians? What are the skills that they should be gathering for both successful research lives, but equally important, practice of medicine life? You've got to be a bit more computationally savvy today than you were decades ago.
Yeah, well, I mean, when I was at medical school, I decided in my third year that I wanted to learn statistics, and you could do a third year in a particular subject. You could do anatomy, physiology, biochemistry, history of medicine, psychology, but not statistics. So I ended up transferring to George Washington University because I'd heard you guys were more flexible. And my medical school wanted to throw me out. They said, what the hell has mathematics got to do with medicine? So my advice would be that it's rather the question is, what has medicine got to do with health? And if I was thinking about medicine now, I would be boning up on informatics, mathematics, because the medicine will come. I learned more medicine after I left medical school from Richard Pito, who was a statistician, than I ever did at medical school.
Yeah, I mean, the way it should be thought of is mathematics is now a foundational science of practicing medicine.
Yeah, I mean, and it used to be that people who went to medicine in Britain were people who couldn't count. Now it's the other way around.
So maybe stop teaching the Krebs cycle and start teaching math and programming.
Yeah, who can remember the Krebs cycle?
All right, well, I want to thank you, Rory, for joining us. Please join us in
Thank
thanking
you.
Rory.